Yeah, recording. Hi.
All right, hello. So my name is Zuzana. I'm a current high school graduate. I've been a researcher for the past 3 years, and I've been working in fields of developmental neuroscience as well as COVID-19 diagnostic test development. And I'm currently an incoming freshman at Stanford University. This talk will focus on the science behind COVID-19 diagnostics and putting some biology right.
Yeah. So I'm Krish, and I lead the Marbella chapter of the Youth Education Initiative. And I'm meeting with Zuzana for learning more about her research on COVID-19 tests. So, um, so yeah, this is some information about Zuzana. Can you elaborate on them?
All right, so all the points are right. I'm an inspiring scientist. I've been working in labs since I was 14. As I mentioned, for first time, I've been focusing on developmental neuroscience, more concretely on stem cells and how they develop into different types of neural tissues. And yeah, as it says, I'm Slovakian, but I've studied in Scotland for the past 2 years thanks to an academic scholarship. I'm also a freelance science writer. I write for a few national newspapers, and I've had an opportunity to collaborate with a young global scientist journal, where I've been a chief editor for science, and I also wrote several pieces on science. Indeed, I'm an incoming freshman at Stanford University, and this year, well, more concretely, last summer, I've interned in an international company called Multiplex DX, which, apart from other diagnostic tests, develops COVID-19 diagnostic tests. And I've had the opportunity to collaborate in a team to focus on development on lab-based COVID-19 diagnostic tests, which I will elaborate on more scientifically later in this presentation.
Perfect. So can you tell us more about your research journey and how it connects with COVID-19 diagnostic tests?
Yeah. So as I mentioned, I started my research journey quite early because very early on in my life, I knew I wanted to become a researcher since I not only like to learn, but I wanted to be on the front lines. I wanted to not only be one of the people that read about information and acquire the knowledge through books. I want to be one of the people who discover new knowledge that people will learn about, which is why I applied to different laboratories in Slovakia. But because I was so young, I struggled to find the right lab. Obviously, there was some prejudice because of my age, so I wasn't accepted at first. But then I found a really open-minded researcher in Czech Republic, concretely in Masaryk University, where I got accepted as an intern for summer at the age of 14, and I gained some lab training, I get some practice and different sorts of lab techniques, lab methods. And I also had the opportunity to work and collaborate on a project related to neural rosettes, which is a 2D neural formation derived from stem cells. A year later, I had the opportunity to have an internship, same laboratory, where we grew cerebral organoids, which are sort of like mini brains, also derived from stem cells. And because of COVID-19, I sort of changed my direction a bit from neuroscience into biochemistry and diagnostics because I felt like I wanted to be an active player in a world where we're trying to combat the pandemic. So I applied and was accepted to an internship at a company I mentioned before, Multiplex DX, where I worked in during summer, and I co-developed the land-based COVID-19 diagnostic test.
Perfect. So what are the different types of COVID-19 tests and how do they differ?
Yes. So there are many different types of COVID-19 tests out there. And of course, it's not that easy to reduce them to 3 types, but I would say that if we're talking about the main types, I would put them into 3 main categories, while under these 3 categories, they also vary and differ depending on how the scientists that developed them designed them. So the 3 main categories I would say are firstly, the antibody test. The antibody tests you can derive from the name is based on detection of COVID-19 antibodies present in patients' blood sample. And it's not as much of a COVID diagnostic test as it is a test for whether someone overcame a COVID-19 infection, since we can detect the antibodies only later after the onset of infection, so not too early on. The second main category I would talk about is lateral flow tests, which is also known as the rapid COVID-19 test, as it is very often used by schools or homes when they need to test someone very quickly. This test is based on a similar principle of antibody tests, as it creates the antigen antibody complex and uses that as a tool for detection of COVID-19. The difference is here that while the antibody test obviously detects the presence of antibodies, lateral flow test does it the other way around, as it detects the presence of COVID-19 antigens present in the patient's nasopharyngeal swab. And it does so, and does so pretty reliably, pretty accurately, but some would argue that, and the data argues and data shows that this test is not as accurate as, for instance, genetics-based tests, which is the third category I will talk about. And this category includes both RT-PCR tests, which is very famously used all around the world, as well as the RT-LAMP test, which is the type I've been co-developing over the past summer. And this third category of tests is based on specifically the detection of COVID-19 genetic information. So it's highly, highly specific. And generally, this category of tests is the most accurate based on the clinical data we've acquired so far.
How long does immunity from through the antibodies last? Like, how long does it last?
Sorry, I haven't heard your question properly.
The immunity through antibodies, like when you have antibodies, how long does it last? Like after you get COVID, how long are you immune to COVID?
It really, really depends. As some people show that they got the immunity, the load of antibodies is present in blood sample for several months. So I think up to that point can be detectable. While the earliest points, I think, where we can detect the antibodies in patient blood is after several weeks from the infection by the virus. But as I said, I've been co-developing LAMP-based diagnostic tests. So the information I've acquired on the other tests is heavily based on reading, not on my personal experimental experience.
Around how long did it take you to develop the test, for example?
Yeah, so firstly, I would like to note that I haven't developed it by myself. I've been more of a help development of the LAMP-based diagnostic test, as the journey of developing the LAMP test started earlier and finished later than I came into the labs of Multiplex DX. So general tests development took several months, while I've been helping the team for around a month and a half approximately.
Which type of test would you use if you were to choose, and why?
Well, it depends on the specific situation. So for me to if you allow me to, I would like to perhaps elaborate more on the science behind each test. Would that be all right?
Yeah.
All right. So, and I will connect that to my reasoning of why would I use different tests under different circumstances. So as I mentioned before, the antibody test is based on the antigen antibody complex formation. So the blood sample is collected from patient, where the liquid part called serum is separated from the blood cells, because the serum contains the antibodies we're trying to detect. And that is put on a test device. And as the sample moves across the test device, if the antibodies are present, it binds to antigens that are in the test device, and that produces some sort of color change, because there's always some sort of conjugated label which allows for this color change to occur. And as I mentioned, as we discussed together, the antibodies are generally present later, in the later part of COVID-19 infection or after the infection. So this test, I wouldn't use it primarily to test for whether someone has COVID. I would use it to test whether someone in the past had COVID, or to discover whether they have the immune reaction for COVID. That's when I would use the antibody test. Then regarding lateral flow tests, again, it's the same principle of antibody-antigen complex, just in reverse. There is a nasopharyngeal swab taken from patient, which is mixed with buffer to create a solution, which is then passed to some sort of adsorption path through which the sample moves along the test device. And as this happens, the antigen, if they're present in the patient sample, they bind to the antibodies, the specific antibodies if they're present in a test device. These antibodies, again, have to be conjugated with some sort of label so that in case the patient sample is positive, it can produce a color change. This test is fairly reliable in the sense that it can test for the protein, the antigen presence of COVID-19. However, as we discussed, as I mentioned before, the problem with lateral flow test is that it's not necessarily that accurate many times. And disadvantages of it is that they often require quite high COVID-19 loads in someone's sample for the results to be detected. So often they're false negative. So someone is positive, but the test evaluates as negative. However, the upside of lateral flow tests is that they're fairly quick and anyone can do it by themselves. So if you really need some really quick tests for some sort of reason, I would definitely go for lateral flow test because it's quick, easy, and it's usually quite easy to access. Then the third category, as I mentioned, is RT-PCR and RT-LAMP test. So PCR reaction is generally quite famous. It's been awarded Nobel Prize in 1990 because it's quite a genius reaction for detection of any sort of pathogen, and it's been widely used in science. And yeah, it's pretty much based on the technique of heating, of putting genetic information into a thermal cycler in some sort of solution. And this solution then the reaction undergoes at different stages at different temperatures. And as that happens, the genetic information is amplified. So a really small load of genetic information can produce many copies of DNA. Or in case of COVID-19, we also need one extra additional step to add on to that, and it's the reverse transcriptase stage, which means that we need a reverse transcriptase enzyme because COVID-19 is a RNA-based virus. So we first need to convert RNA into DNA, and then we can amplify the genetic information. And this is a very, really accurate technique. But as I mentioned, the problem is that for that, you require thermal cycler. You require different temperatures for different stage of the PCR reaction. The PCR reaction has three main stages, which is the denaturing, where DNA unwinds. Then you need annealing, where the primers, short strands of DNA, binds to the single strands. And then the first stage, which is the elongation, which is where different nucleotides are added onto it and like DNA is replicated. And each of those stages, as I mentioned, requires different temperatures. And for that, you need quite heavy equipment. And generally, the assay is quite complicated and it can take up to several hours for a result to occur. Therefore, yeah, that's the disadvantage of PCR is it's very long. So if I needed a quick test, I wouldn't go for PCR. But if I wanted a really, really accurate test, I would definitely go for RT-PCR. On the other hand, RT-LAMP, which is very similar to RT-PCR, kind of combines the benefits of lateral flow tests and RT-PCR tests, because LAMP test is based on similar principle as RT-PCR, so it detects genetic information, so it's highly accurate. However, because it's isothermal, which means that it happens on one single constant temperature, you don't require the thermal cycler. So it's less complicated, it also takes less time. It usually takes up to like 30 minutes. Therefore, if I wanted a test that's both very quick and very accurate, I would go for RT lamp test. So yeah, these are different situations.
Yeah. So what do you think are the common hoaxes surrounding COVID-19 diagnostics? Could you try to debunk some of them?
All right, they're probably different in different countries. I came across different hoaxes when I talked to my friends. And when I'm talking about Slovakia, for instance, the country where I come from, the country where I live, one of the most common hoaxes is that the RT-PCR test is detecting the presence of genetic information of humans, and thus companies are acquiring information about our genetic code and things like that. People are afraid. So to debunk that, I would just say that RT-PCR test for COVID is designed to detect genetic information, COVID specifically. It's not detecting the presence of genetic information of any other organism, nor our human DNA. So it's just not true. It's detecting, it's very, very specific for COVID-19 genetic information. It's designed like that, and it's meant to detect that. So that's what it's doing. It's not detecting your personal genetic information. Another quite ridiculous myth I heard of is that some people believe that if they will drink alcohol, they do not need to get tested for COVID because the alcohol will kill the COVID particles, which is, yeah, it's quite ridiculous. But let's approach it scientifically, and let's just say that yes, it's true. Alcohol is often used as disinfectant on surfaces. Even when you work in labs, you use alcohol to disinfect the surfaces to prevent for contamination. But when we're talking about consuming alcohol, it has no link to killing the viral particles within you. Therefore, it won't kill the viral particles. Thus, you still need to get tested if you want to go somewhere and you want to find out whether you're positive or negative or something like that. Still test. Alcohol, if you're positive, alcohol will not kill COVID-19 nor any other viral particles within your system. Because that's a common misconception I heard of, and it has really no scientific basis. The other thing I heard of regarding, it's more about getting rid of COVID than about COVID-19 diagnostics. But many people believe that, similar to the alcohol point, they believe that if they're gonna shine UV light on themselves, they're gonna kill all the viral particles in themselves. And again, it's similar to the previous point. UV lights can be used to disinfect surfaces. It's not used to cure yourself from, for instance, COVID-19. Therefore, it's not like a precautious measure you should take. If you want to travel somewhere, go somewhere, either if you're not vaccinated yet, or even if you are vaccinated, go and test yourself for COVID-19. It's the safest thing and the most rational thing you can do.
And the worst thing is that people sometimes in power, they are the ones who are spreading the misinformation.
Yeah, definitely. Yeah, and definitely my biggest advice to people is that when you're reading something, always check what's the source of the information you're reading, who wrote it, or what's the source of your source. Did they look up the data that's scientific, or is it just something someone made up? It's really important to check your source.
How do you think you can reduce the spread of misinformation and educate people about?
Definitely read from several sources, get your information from several sources, and always think about what you're reading. If something sounds really extreme or not heavily based on scientific reasoning or scientific data, always try to check where does the information come from. And educate yourself, find different sorts of applications, different sorts of organizations who are known to promote correct information, who are known to debunk the misinformation. And lastly, discuss the information you've acquired, discuss it with different people so that you can sort of fact check each other and come to a conclusion whether something is a legitimate information or whether something is a misinformation.
And about like, it's not related to tests, but like COVID vaccines, what do you think about misinformation about them? Because there are a lot of people who are skeptical about it.
Yeah, I mean, I've been quite frightened by the amount of misinformation I've heard regarding COVID-19 diagnostics, but it's even worse when it comes to COVID vaccines. And yeah, I think the problem here is that with vaccines, there's a massive element of human fear playing in. So when people are afraid, they're very, very likely to be victims of their own confirmation bias. So they're afraid of something and then they read data, or not even data, just an article without proper scientific data that fits this picture of what they're afraid of, and they'll use it as their reasoning for having this fear. And this fear then prevents them from getting vaccinated by legitimate, clinically validated COVID-19 vaccines. But we need those vaccinations because we need to reach the herd immunity. So I always say to people who are doubting whether they should go for a COVID vaccine, the risk in not getting vaccinated is much higher than the risk in getting vaccinated.
What's your opinion about like the new variants of COVID, right? So how do they differ from the previous ones? Why they worse or like, yeah.
Yeah, so the Delta variant you're talking about.
Yeah.
Yeah, the Delta, yeah. So from what I've read, this is, yeah, as I said, I'm not a trained virologist nor trained epidemiologist. I'm just someone who worked a bit in science and likes to read a lot. And from what I've read, it seems like the problem with this variant is that it is highly transmissible, which means that it's really, really easy for this variant to infect a lot of people very quickly, which is quite dangerous because when we're talking about viruses, we generally talk about two main properties: transmissibility and virulence. Transmissibility is, as I said, it discusses how many people a virus can infect, how quickly. Whereas virulence discusses how harmful a virus can be to its host. And usually it happens to be so that it's much more strategic for a virus to be highly transmissible and not virulent, because if a virus kills the host or kills the person too quickly, it means that it cannot spread as much. However, if it's very virulent, if it's very transmissible, it means that it doesn't kill the person much, but it can transmit to many people quite quickly and thus replicate much more quickly. And yeah, the Delta variant, from what I've gathered, is highly transmissible.
Yeah. So also, could you elaborate more on why chemically the rapid tests are less effective than the other kinds of tests?
As I discussed, when we talk about rapid tests, we generally talk about lateral flow tests, which, as I said, are based on detecting the antigen of COVID-19, like the protein that's present of COVID-19. And this test, although I would still take it when necessary, it's firstly, it's less accurate than PCR tests because clinical data has showed so. PCR and generally genetic information-based tests are more accurate than tests just based on detecting the antigen of COVID-19. So if you want an accurate information, it's more productive to either go for RT-PCR or RT-LAMP test. While another factor why I think that rapid tests are considered less effective is because, as I mentioned, I think before, is that it is required to have quite a high load of COVID-19 within your sample for the lateral flow test to detect the presence of COVID-19. Whereas in case of, for instance, RT-PCR tests, you require quite a low viral load for it to amplify the genetic information and detect its presence. So yeah, these are the two main reasons I would say.
How effective do you think masks are in controlling COVID-19 spread?
Masks. Masks are definitely very, very important factor in preventing the COVID-19 spread. I'm not too aware of like the whole mechanism of the barrier, of how the mask barrier works, but clinical data has shown that the risk of infecting someone or getting infected by COVID-19 is much, 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 much higher if people are not wearing masks. So for the highest effectiveness of preventing the COVID-19 spread, all the parties, so every person you're talking to should ideally wear a mask. Even if you have a group of 5 people, for instance, if even a single person is not wearing the mask, there's already a higher risk of some sort of infection happening. So yeah, it plays a really vital role in prevention of spread.
So to conclude, what message do you have for the people who are watching this?
All right, so definitely get tested is required. All types of tests have their own benefits, and yeah, depending on what your situation is, discuss with your doctor, discuss with some professional which test is the most suitable for your specific situation. And definitely get vaccinated if it's possible. Again, the risks of not getting vaccinated are much higher than the risks of getting vaccinated. Wear masks, and yeah, be careful around misinformation. Read a lot, check facts, discuss facts with people, and to learn what's the actual truth and what's the misinformation. That's my final message.
Yeah. And which vaccine do you think would be the most effective? Because there's like the Pfizer, the AstraZeneca, and yeah.
Yeah, it depends, but the clinical data seems to show that Pfizer vaccine seems to be so far the most effective.
Yeah.
From what I've
read.
Thank you. No problem. Thank you very much for inviting me.
Yeah. So this was how COVID tests work.